Are you a biology student? And are you confused with the concept of mitosis? Well, you are at the right place. I am Professor Miithyl Dave and I welcome you to MD's World of Concept. In this particular video, I am going to make you understand the concept of mitosis and also its various stages. So let's get started. Alright, so let's get started and understand the mitosis and its stages. So let us first understand what is mitosis. So mitosis is a type of cell division in which one cell, that's the mother cell, divides to produce two new cells, also called as daughter cells, that are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. The major purpose of mitosis is for growth and to replace worn out cells. If not corrected in time, mistakes made during mitosis can result in changes in the DNA that can potentially lead to genetic disorders. So let's understand the various stages of mitosis. These are the stages of mitosis. So we have the interphase, the prophase, the metaphase, the anaphase, the telophase and the cytokinesis. Now when we talk about interphase, prophase, metaphase, anaphase and telophase, it's known as karyokinesis, and then what happens is my cytokinesis. So we're going to learn and understand each and every stage in detail. However, before that we need to understand what's a cell cycle. Cell cycle is basically an actively dividing eukaryotic cells pass through a series of stages known as collectively cell cycles, which is nothing but two gap phases, G1 and G2, and S phase for synthesis in which the genetic material is duplicated, and then there is an M phase in which the mitosis partitions the genetic material and the cell divides. When it comes to G1 phase, metabolic changes prepare the cell for division. At a certain point, which is known as the restriction point, the cell is committed to division and moves into the S phase. When it comes to the S phase, the DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. In the G2 phase, metabolic changes assemble the cytoplasmic materials necessary for mitosis and for cytokinesis. When it comes to the M phase, the nuclear division known as the mitosis followed by a cell division known as the cytokinesis. The period between the mitotic division, that is G1, S and G2, is known as the interphase. Let's understand the cell cycle with the help of this diagram. So initially we have the G1 phase, then we would have the S phase, then we would have the G2 phase where the cell will then undergo the preparation for mitosis. Then these are the four ones P, M, A, T, PMAT, known as prophase, metaphase, anaphase and telophase. And once telophase is done, it will undergo cytokinesis and then it will start again with the new cells. So let's now understand interphase. In interphase, the DNA in the cell is copied in preparation for cell division. This results in two identical full sets of chromosomes. You can see in this particular diagram, outside the nucleus are two centrosomes, each containing a pair of centrioles. So you can see the centrioles over here which are in two pair. These structures are critical for the process of cell division. It is only when the centrioles are present that is the time when the cell understands that it is time for division. During interphase, microtubules extend from these centrosomes. Then let's understand prophase. The chromosome condenses into X-shaped structures that can be easily seen under a microscope. You can see the purple ones over here. Each chromosome is composed of two sister chromatids containing identical genetical information. The chromosome pair up so that both copies of the chromosome 1 are together, both copies of chromosome 2 are together, and so on. At the end of the prophase, the membrane around the nucleus in the cell dissolves away, thereby releasing the chromosomes. It is very much important that at this stage the nuclear membrane has to go away. The mitotic spindle, consisting of microtubules and other proteins, extends across the cell between the centrioles as they move to opposite poles of the cell. Now let us come to the later part of the prophase, also called as the late prophase, sometimes prometaphase. The mitotic spindle begins to capture and organize the chromosomes. So basically this is an intermediate stage between the prophase and the metaphase. This is how the entire diagram looks like. The chromosome finish condensing, so they are very compact. The nuclear envelope breaks down, releasing the chromosomes. The mitotic spindle grows more and some of the microtubules start to capture chromosomes. Then we go ahead to the metaphase. In metaphase, the chromosomes line up neatly end to end along the center, also called as the equator of the cell. How you can see over here, the purple color chromosomes are lined at the center of the entire equator. On imaginary lines, these are called as the spindle fibers. The centrioles are now at opposite poles of the cells. One you can see on the top, another at the bottom, or maybe on the north pole and the south pole, with the mitotic spindle fibers extending from them. So you will be able to see there's going to be certain lines, right, known as the spindle fibers, which are attached from the north pole to the south pole. The mitotic spindle fibers are attached to each of the sister chromatids. You can see in the diagram again. So before proceeding to the anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate with their kinetochores correctly attached to the microtubules. This is called as the spindle checkpoint and helps to ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If the chromosome is not properly aligned or attached, the cell will halt the division until the problem is fixed. Now let's go ahead to anaphase. In an anaphase, the sister chromatids separate from each other and they are pulled towards the opposite ends of the cell. So basically they start proceeding towards going to the either side of the poles. The protein glue which holds the sister chromatids together is broken down, allowing them to separate. Each is now its own chromosomes. The chromosome of each pair are pulled towards the opposite end of the cell. The microtubules not attached to the chromosomes elongate and push apart, separating the poles and making the cell longer. All these processes are driven by motor proteins, molecular machines that carry or can walk along microtubule tracks and carry cargo. In mitosis, motor proteins carry chromosomes or other microtubules as they walk. Then we come to the telophase. In telophase, the cell is nearly done dividing and it starts to re-establish its normal structure as cytokinesis, which is division of the cell content, takes place. So you can see in the diagram, the mitotic spindle is broken down into its building block. The two nuclei form, one for each set of the chromosomes. The nuclear membrane and the nucleoli reappear. The chromosomes begin to decondense and return to their stringy form. And the last one is cytokinesis. Cytokinesis, the division of the cytoplasm to form two new cells, overlap with the final stages of the mitosis. It may start in either anaphase or telophase depending upon the cell and finishes shortly after telophase. So you will be able to see there is an animal cell and there is a plant cell, and both of them have two different processes. In animal cell, cytokinesis and contractile pinching the cells in two like a coin purse with a drawstring. The drawstring is a band of filaments made up of protein which is called as actin, and the pinch crease is known as the cleavage furrow. You will be able to see the cleavage furrow over here. The plant cells can't be divided like this because they have a cell wall and they are too stiff. Instead, a structure called as a cell plate, which you can see over here between the two cells, there is a cell plate over here, forms down the middle of the cell, splitting it into two daughter cells, separating by a new wall. So students, I hope you understood the concept of mitosis and its various stages. If yes, do share this video to your friends and also give me a like on this video. Do subscribe to this channel and leave in the comment below what was that one stage that you liked the most and which you felt is the most important. This is Professor Miithyl Dave signing off.